All right, let's talk about analysis of covariance. So analysis covariance, or ANOVA, when do you use this thing? Well, when you have one nominal between subjects independent variable, it's kind of two or more levels, and you've got a dependent variable that's measured on either an interval or a ratio scale. And that would just make it ANOVA. But in this case, you want to adjust for the potential confounding effects of one or more covariates, so other variables that might otherwise mess up your study, confounders, if you didn't adjust for them. So, for example, let's say you want to compare the effectiveness of a county program and a private program for drug treatment, and you want to see how they affect subsequent drug usage. However, there are different types of users that enter the two programs, and you can't randomly assign them. So let's say you wanted to equalize them, for example, on their baseline level of drug use for comparison purposes, that is, statistically adjust them as though they had the same starting level of drug use, and then see if the programs are equally effective or if one is better than the other. Your independent variable in this study would be the type of drug treatment program. That would be county or private. Your covariate would be their prior drug use, that is, what their level of drug use was when they entered the program, or baseline, and that is measured on a 0 to 10 point scale with higher numbers indicating more drug use, that is at baseline. Your dependent variable is also drug use, but it's post-treatment drug use. So let's say it's drug use on that same scale, 0 to 10, higher numbers equal more drug use. So continuing that example, let's say that you obtain that is baseline drug use on that 10-point scale, and 6 months post-drug use, that is for folks after treatment. 6 months later, for 50 clients at the two counseling centers, there's 25 at each counseling center. And assume that which is probably true, the more hardcore users, that is those who had the bigger drug issues, tended to be remitted to the county program, not fancy private programs. So let's say these are the post-treatment means that you get in drug usage. Again, a higher number indicates more drug usage. Looking at this, post-drug usage, the county treatment programs are 5.36, private treatment programs are 3.52. Sure looks like the county programs aren't as effective as the private programs. Their mean drug use is higher after treatment. So looking at just those post-treatment means, it looks like the county programs are more effective, but this ignores the fact that the county programs tend to get more serious users. So the county patients were worse from the get-go. That is, those who go to county have higher drug use from the start. So the fact that they have higher drug use after doesn't necessarily mean the program didn't work. It could just be that they started higher. All right, well, what if we had collected the baseline drug usage, like I said we had in this example? We could see the potential bias that the more hardcore users go to county. So if we could take into account that pre-existing difference in drug use between the county and private programs, our conclusions about program effectiveness might differ. So let's say here's the data. If we now look at the pre-drug use versus the post-drug use, you can see drug use did go down in both the county and the private programs. And in fact, if you do the difference, it went down even more in the county programs. That is, it went from 6.76 to 5.36, which is a 1.40 drop in subsequent drug use. Versus at the private programs, it went from 4.48 at baseline to 3.52, which is just about a 1 point drop in drug use. Even though the groups differed in their pre-existing drug use levels, it does look like the county program is somewhat more effective. It resulted in a larger amount or a larger reduction in drug use than the private programs. So, getting back to ANCOVA, if you looked at just post scores, the private program looks more effective just because they had less hardcore users in the first place, right? So the fancy treatment program that you pay a lot of money for, well, tends to be folks who have other means, and the less hardcore users tend to go there. So the county program just looks worse from the get-go. It has potentially confounded by prior drug use level. So the point of doing ANCOVA in this example is that you would attempt to statistically equalize the groups on their prior drug use level before determining if they differ on their subsequent drug use level. That is, you statistically equalize them. And in this sense, ANCOVA first does a linear regression where it sort of adjusts for covariates, and then it does the equivalent of a ANOVA or a t-test in this case, where it says, okay, after I adjust, do these means differ? So you have what are called crude or unadjusted means and adjusted means. So let's talk a little bit about covariates. Covariates are the variables that you want to adjust to remove the differences associated with the dependent variable and the independent variable, technically, prior to determining the effect of the independent variable. The covariates are the things you want to remove the effects of from that dependent variable. Typical studies that we do, for example, dissertations, are quasi-experiments, and the researchers don't really get the opportunity to randomly assign, which would be expected to equalize the groups on both known and unknown variables that are related to the independent and dependent variable. We're stuck with quasi experiments. You know, if you're doing a dissertation where you work, you are probably going to be stuck just comparing folks who went through one type of therapy versus another, or longer treatment, shorter treatment, etc. You're not going to get to randomly assign. So if you can't randomly assign, the next best thing to do is to try to statistically equalize the groups on those factors that are known to be important. However, you don't want to go crazy and get too many covariates because that has its own sort of problems. So if a covariate is related to the outcome and the distribution of the covariate differs between the independent variable levels, then it is what we call a potential confounder in your study. It's a variable that could confound the relationship between the independent and dependent variable. So it's important, so I'll say it again, a variable must be related to both the independent variable and the dependent variable for it to be a potential alternative explanation for the differences that you observe between the independent variable groups on the dependent variable. So covariate, to be a confounder, has to differ between the groups in its distribution. And so in this case, prior drug use would have to differ between the groups. And also, it would have to be related to the dependent variable. And do you suspect that prior drug use is related to subsequent drug use? Probably. That's the way life works. So let me give you an example. So the early studies of driver's education were quasi-experimental. They basically compared teens whose parents made them go through driver's education to teens whose parents didn't make them go through driver's education. And they found that there's a huge benefit of taking driver's education on teens' subsequent crash risk. That is, those who went to driver's education tend to have much lower crash rates than those who didn't, even though everyone got licensed. However, it turns out that conscientious parents were more likely to send their kids to driver's education, and those conscientious parents are also the ones who are like asking their teens, like, who you going out with, how late are you going to be staying out, you're not going to be drinking. They tend to monitor their child's driving behavior, which also lowers crash risk. So you have what we call a classic confounding triangle here. You're trying to, and so exposure here is the independent variable, outcome is a dependent variable. You're trying to estimate the relationship between your independent or exposure and your outcome variable. In this case, that's driver's ed and teen crashes. You think that they're related. However, you have this third variable or confounder that's causing a problem, that is parents who care. So parents who care both put their kids in driver's ed, and parents who care also monitor their teens to ensure that they don't crash too much or do things that are associated with increased crash risk. Caring parents is a confounder in this example. So it turns out later studies, they actually did a gigantic true experiment in Georgia where they randomly assigned teens to one of three groups: normal driver's ed, the best driver's ed that the driver's ed companies that could come up with. It was like a really long program, or no driver's ed. And when you, so meaning they literally said, you don't have to take it, go get your license. Whereas the other two had to go through driver's education. When you do that, it turns out there is no long-term benefit of driver's education. That is, after 6 months, crash rates are basically the same for all the groups. So the true experiment removed that effect of confounding, that is the caring parents, by using random assignment. So a little bit more about covariates. You can have more than one of them in a study. What you should do before you go throwing a bunch of covariates into an ANCOVA is establish a couple things. One, that your independent variable groups do indeed differ on each of the covariates. So if it's average age, for example, the average age of your groups is different, if you think age is a potential confounder. But that's not enough just to have average age differ. Average age, for example, would also have to be related to the outcome variable. So the second part of this is that the covariates are related to the dependent variable. So two things have to be true for something to be a confounder. It has to differ between the groups, and it has to be related to your outcome variable. A third piece, just to throw on top, this is if you have more than one covariate in a study. You don't want covariates that overlap too much, ones that are too strongly related to each other, because otherwise they're basically redundant. You don't need all of them. So that's the third piece. Related to the, or differs between the levels of the independent variable. It's related to the outcome, and if there's more than one covariate, they don't overlap too much. So what sorts of preliminary analyses should you do to establish that a potential covariate is a confounder? Well, one thing is to run Pearson correlations between your dependent variable, independent variable, and your covariate or covariates. If there are multiple covariates, this will show you how strongly overlapping they are as well. The other thing to do is to make comparisons of average covariate scores or covariate distributions between the independent variable groups. That is accomplished in the case of a categorical covariate. For example, if you're adjusting for gender as a covariate, you would use a chi-square test of independence to determine if the independent variable groups have different distributions of that categorical covariate. That is, does the distribution of gender differ between the levels of the independent variable? If you've got a continuous covariate, something like age, you wouldn't use chi-square. You're going to be comparing the mean age between levels of the independent variable. That is an independent samples t-test that you would use to do that. You want to be able to use that test to determine is the average of the covariate different between the independent variable groups. So again, covariates are only potential confounders in your study. And don't get me wrong, you can throw covariates in, but they're only potential explanations for your effect if the covariates are first related to the dependent variable. So that would be indicated by Pearson correlations between the covariate and the dependent variable, and the distribution of the covariate differs between the independent variable groups. So that would be indicated by either the chi-square test of independence or independent samples t-test that compares the mean covariate levels across the independent variable. If those two things are true, then that covariate can explain differences between the independent variable groups on the dependent variable. It's like caring parents in the driver's ed and teen crash rates thing. If it doesn't meet both those criteria, it cannot be a confounder. You can still adjust for it. So again, just to emphasize it, because this can help you during your dissertation defense, if either of the above is not true, then the covariates cannot be confounders in your study. So hair color may differ between the groups, but if hair color is not related to self-esteem or whatever your outcome variable is in your study, hair color cannot be a confounder. Continuing with covariates, there are three possible outcomes that can result if you adjust your dependent variable for covariates in ANCOVA. The first is nothing. You adjust for things, you throw that variable in as a covariate, and nothing really happens. The means don't really change that much. You can still reduce the error, which is not a bad thing, but you don't really see much change in the means. Another thing that can happen is that you see the means get much closer together. So that suggests that the covariate did remove confounding. So if the means get closer together, after you put the confounder into the ANCOVA, it tells you there was some sort of confounding there, and it gets adjusted by moving the means closer together. The opposite of that can also happen. The means can get further apart. So you adjust for age and sex, and next thing you know, the means get further from each other. So those adjusted means. So that tells you that, again, confounding was removed by adjusting for those covariates. So there are multiple effects involved in the ANCOVA. It looks a lot like ANOVA, to be honest with you, except that there's extra lines for each covariate. So the covariates are first used to adjust the means through regression analysis. That happens automatically, like you don't even know it's happening. You just run an ANCOVA in SPSS. After it adjusts the means using multiple regression, it then does an ANOVA, comparing the dependent variable adjusted for the covariates, whether it differs across the levels of the independent variable. So this is all sort of opaque to you. You don't see it happening, but that's what's happening. It actually first adjusts the means and then says, okay, after I take into account these covariates, I adjust them, the dependent variable means, is there a difference between the independent variable groups on the dependent variable? So you get a linear table for each covariate. Now, covariates, you will get a sig value that you can determine if it's statistically significant. Even if the covariate's not statistically significant, the means could still be adjusted somewhat, or it could reduce the error variance. So including covariates can still have a positive effect, even if they're not potential confounders. And you can use covariates in designs that have more than one independent variable, which we'll get into soon. And you can have more than one covariate. So incidentally, if the covariate is significant, or covariates, it tells you that they did significantly adjust the means. If they are not significant, it tells you they didn't really meaningfully adjust the means, but they could have other beneficial impacts. So in the case where you have more than one independent variable, you will get main effects, interaction effects, but they're all adjusted for the covariates in the model. So let's talk about the sources of variability in ANCOVA. So here you can see the bubble for the dependent variable is all the variability associated with your dependent variable in your study. Now, some of that variability in this ANCOVA can be accounted for because it's the relationship of the independent variable directly with the dependent variable. Now, some of that variability is on the bottom there, what the covariate predicts in the dependent variable. And then some of the variability could be predicted either by the covariate or the independent variable. And so you've got multiple sources of variability, unique variability predicted by the independent variable, unique variability predicted by the covariate, and then overlapping variability that could be attributed to either the covariate or the independent variable. So assumptions of ANCOVA. Now, because ANCOVA is both a mixture of ANOVA and linear regression, it's got all the assumptions of both of those. So first of all, it's independence. That is, you've got a between groups design, different people, and the levels of the independent variable. So that's independence, which we've talked about before. Number two, we've also talked about normality. The scores in each cell of the design or each level of the independent variable are approximately normally distributed, and you can test for that using the explore procedure. However, we're looking for skewness, and if our skewness coefficient is 2 or larger from SPSS, and even if you do violate it somewhat, that is, you've got a skewness coefficient that is absolute value of 2 or bigger, It turns out that it doesn't really mess up the ANCOVA that much because ANCOVA, like all ANOVAs, is pretty robust to minor to moderate deviations from this assumption, but it's still good to test for. Homogeneity of variance across all cells in the design or all levels of the independent variable. So this is the same as it's been in the past. The spread of the dependent variable scores is approximately the same for each level of the independent variable, or in the case of multiple independent variables, for each cell in the design. So normality was the scores for each level are normally distributed. This one says the spread is about the same across the different levels. So how do you test this? Well, you click homogeneity test in SPSS and you get Levene's test. If you violate this assumption, the result is that you have type 1 error inflation. And so what do? Do you give up and do nothing? No, one option is to use a tighter alpha level, so 0.01 for example, instead of 0.05, in an attempt to decrease the amount of type 1 error inflation from violating the homogeneity variance assumption. Next assumption is absence of multicollinearity in singularity, which I don't think you've heard of. I don't think we've covered yet, but basically this says that if you have more than one covariate in the model, they're not too strongly related, so they're not completely or mostly overlapping. So rule of thumb for that is the correlation coefficient, the Pearson correlation between covariates. Any two covariates is not 0.5 or higher. So that would suggest that there's a lot of overlap between the covariates, and maybe you don't need them all. So the assumption here is that you don't have too strongly correlated covariates in your model. If they are too high, what happens? Well, SPSS crashes, so it can't converge on a solution. So again, high correlations indicate that there's probably redundancy in your covariates, and maybe you don't need them all. Then we have linearity. So so far we've only covered linear regression, and so that means it uses a straight line as the relationship between the covariate and the dependent variable. And so in that sense, it needs a straight line to do a good job of representing the relationship between the covariate and the dependent variable. So that is for each covariate and the dependent variable, and each pair of covariates should approximately have a relationship that can be well represented by a straight line. That is what linearity means. If you violate this, you lose power in your analysis. That's the big effect. How do you check this? Well, you do a bunch of scatter plots. So we've done scatter plots in the past. You do a scatter plot between each covariate and the dependent variable, and then each pair of covariates. I'm not going to make you test this here, but that is how you would test it if you ever wanted to test this assumption or asked to by your committee. So the homogeneity of regression slopes, or the equal slopes assumption, is the big one that people talk the most about ANCOVA. And this basically says that whatever regression line it comes up with, so makes a linear regression line, whatever that regression line is, that it's about the same for each level of the independent variable or cell in the design. So it uses a single linear regression equation to adjust all the cells for the covariates, or all the levels of the independent variable for the covariates. So the assumption here is that a single regression line is does a good job of adjusting across the levels of the independent variable. That is, the regression equation is the same for each level of the independent variable or cell in the design. And that's what I say here. So ANCOVA only uses one regression line to adjust all the dependent variable scores. So if the relationship is different for different groups, so it's negative for one and positive for another, you've got a problem. So you won't get the best possible adjustment, and you could have residual confounding, which suggests you'd have power issues. So how do you check this? Well, you can do a split file for each level of the independent variable, and you can compare regression coefficients, that is run regression, of the covariates on the DV for each level of the independent variable and see if you get similar equations. If you get stuck doing this or checking this for your committee, you probably want to hit me up. So in analysis of covariance, you have both what we call adjusted or crude means and, unadjusted or crude means and unadjusted means. So we need to talk a little bit about them. So adjusted means take into account any relationship between the covariate and the dependent variable. They are adjusted for that relationship statistically. This is what we mean when we say statistically equalizing groups. The unadjusted means are just the crude or unadjusted means without any of the covariate adjustments. So they are the same means that you're used to seeing in analysis of variance, without any fancy linear regression to adjust means for the effects of covariates. In SPSS GLM univariate, which is what we're going to use for doing ANCOVA in this class, you have to request the estimated means in order to get the covariate adjusted means. If you under descriptives, you get the raw or crude unadjusted means to get the adjusted ones that show the effect of the covariates adjustments. You need to make sure to request estimated means. So again, the means you get from descriptives is just the unadjusted means. They're the ones you're used to seeing, but in this case, you got to make sure to get estimated means. It is common practice to present both the crude or unadjusted means and the adjusted means as like a figure so that folks can see the effect of what your adjustments did to the dependent variable. So did they get closer together? Did they get further apart after you adjusted? So you show before and after basically adjustment. So when you get the adjusted means from the estimated means, unfortunately SPSS does not give you the standard deviations, but APA requires you to report the standard deviations for those means, those adjusted means. And so it gives you something called the standard error, which I've purposely not talked much about because you don't care. But basically, I just need you to know, how do you convert a standard error of mean to a standard deviation? You multiply the standard error in the estimated means table by the square root of the sample size for that level of the independent variable or cell. So in math, you multiply the standard error that gives you by the square root of the sample size for that group. So not the whole study, but just that group. So, if a group size was, say, 25, square root of 25 is 5, so you'd multiply the standard error by 5 to get the standard deviation. So that's how you convert it. In this case, again, n in this formula indicates the number of participants in that particular group. So it's not hard, but you do have to bust out your calculator or phone for this. Decision rule for ANCOVA. Well, for the covariate, if your ANCOVA sig for the covariate is less than 0.05, then that tells you that the covariate did significantly account for a meaningful amount of variability in the DV. Another way of saying that is if sig for the covariate is less than 0.05, then that covariate did meaningfully adjust the means. If the ANCOVA sig is greater than 0.05, it tells you that that covariate did not meaningfully adjust the means. So less than 0.05, it did some significant adjustment. Greater than 0.05, covariate didn't do much. For the independent variable, if your ANCOVA sig for your independent variable is less than 0.05, then you're going to reject the null that all groups are equal on the dependent variable after adjusting for the covariate. So again, the IV test here is after adjustment, so if any. And so it tells you, this differs from ANOVA, and it's not just do the groups differ on the DV, it's do the groups differ after adjusting for the covariates on the DV. If your ANCOVA sig, on the other hand, is greater than 0.05, it tells you after adjusting, there's no significant difference between the groups on the independent variable. Multiple comparison tests for the independent variable. If your independent variable in your ANCOVA has 3 or more levels, you would need to use a multiple comparisons test such as Tukey, just like for ANOVA, to determine exactly which levels of the independent variable differ from which other levels on that adjusted dependent variable. So if you just have 2 levels, you're basically using ANOVA to run a t-test. You don't have to go ahead and do the Tukey, but if you do have 3 or more levels, it's the test of the independent variables, just an omnibus test that tells you, okay, do any of these groups differ from each other? If it's significant, the answer is yes. You got to follow it up with Tukey to find out which ones differ from which. And the Tukey will compare those adjusted means. Measure of effect size, it's eta squared. Eta squared tells you what it always tells you, the proportion of variability in the dV that can be accounted for by the independent variable. It can range from 0 to 10, or 0 to 1, sorry. Higher scores indicating a higher proportion of the variability. But do note that in this case, it is the proportion of variability in the adjusted dV, so that is what's left after the covariate takes its a portion of variability out. Is there what proportion of remaining variability is associated with the independent variable? There's your formula. It is the sum of squares for the independent variable divided by the sum of squares total. But note that the sum of squares total, you have to hand calculate it. So don't use the one that comes in the SPSS table. Sum of squares total is just the sum of squares for the independent variable plus the sum of squares error. Not included in there, what's not included in there is sum of squares for the covariate. So note that you must hand calculate that sum of squares total as the sum of squares independent variable plus sum of squares error, because you don't, I don't think still you get the correct number in SPSS, so just always hand calculate it. So do not include the covariate in your sum. Note, it's not sum of squares IV plus sum of squares CV plus sum of squares error. The covariate is left out. So again, what this essentially is going to calculate for you is the proportion of variability remaining after the covariate adjustments that is accounted for by the independent variable. APA format for ANCOVA as well. No one does this. They always report these things in tables, and you're going to report it in an ANCOVA summary table as well. I'll show you how to do that at the end. All right. Are you ready to go ahead and do an example of an ANCOVA? Well, shocker. The example we're going to do is the effect of drug treatment on subsequent drug use. So in this case, our hypothesis is there a difference in post-treatment drug use between persons who attend county versus private programs after adjusting for pre-existing differences in drug use, that is their baseline drug use levels. How do county versus private programs compare? Your independent variable is type of drug treatment program, county versus private. Covariate is going to be their prior drug use or baseline drug use. It's measured on that 0 to 10 scale, higher numbers, more drug use. Your dependent variable is 6 months post-treatment drug use, also measured on that 0 to 10 point scale. So this is what the data setup looks like. There's a column for the independent variable, column for the covariate, and a column for the dependent variable. And because our independent variable is nominal, it's just which type of program you went to, we had to use dummy coding, and that is program 1 is county, program 2 is private. So 1 is county, 2 is private. This is important later. Continuing our example, what I first want to do is do an ANOVA, so that is no covariate, just to show you how ANCOVA works. So we're going to compare the post-treatment drug use of those going through the county and private programs without any covariate adjustment, without adjusting for their baseline drug use levels, just to show you how it changes after we do adjust. So this is going to be an ANOVA comparing the treatment groups on the dependent variable, that is post-use drug use, without adjusting for the covariate, which is pre or baseline drug use. Question we're asking here: does post-treatment drug use of county versus private program participants differ without adjustments? There's our means. So county was 5.36 after treatment. Private was 3.52. These are the same means we were looking at earlier. Here's the result of the ANCOVA. You can see there's a row for program and a row for error. So program represents the effect of the independent variable. Our rule is to look at the sig and see if that sig is less than 0.05. If so, we're going to say yes, there's a difference between the county and the private programs in their subsequent drug use. And indeed, 0.001 is less than 0.05, so there is a difference between county and private. What's that difference? Well, look at the two means. Its county was 5.36, private was 3.52. We know those two means are significantly different. Higher means more drug use. So who had higher subsequent drug use? Those who went to the county program. So this is according to this, it looks like the county program is less effective for treating drug users. But we know that the more hardcore users tend to end up in county anyway, and so that this is not necessarily a fair comparison. It's biased potentially by the fact that more hardcore users go to one program over another. So could this finding that the county programs are less effective, that would be your conclusion, than private programs, could this finding just be confounding due to pre-existing differences between those who go to the two programs? I don't know. Let's do an ANCOVA. So now we want to see if our potential covariate, that is prior drug use or baseline drug use, is not just a covariate, but also a potential confounder of this relationship. So in this case, we're going to use an independent samples t-test with the IV being the drug treatment program and the covariate being the DV, that is their baseline drug use. So we're asking the question here, does mean baseline drug use differ between those who go to county and those who go to private programs? That would tell us that the covariate is related to the independent variable. If the covariate had been continuous, something like sex, we would use the chi-square test of independence for this instead, but we do have continuous interval or ratio covariate, so we're going to go ahead with the independent samples t-test. Do the county and private patients differ on pre-treatment drug use? There's our means. So pre-treatment for county was 6.76. Baseline or pre-treatment for private was 4.48. That 2+ point difference between the drug use levels at baseline for those who go to the different programs, is that significant? Well, that's why we do independent samples t-test. Remember, this is the one that has a pre-test on it, that Levine's test that tells you if you read the top row or the bottom row. If the sig for Levine's test is less than 0.05, it means we violated the equal variances assumption. We got to drop a row. If the sig for Levine's test, that first sig there, is greater than 0.05, we can basically assume equal variances. In this case, it's 0.308, so we're good to go. We'll continue assuming equal variances. So then we look at the sig for our test for equality of means. Technically, this is a two-tailed test. We just want to know, do they differ, which is a two-tailed hypothesis. And sig is less than 0.05, which tells us, yes, those two means differ. And in fact, our conclusion, using alpha of 0.05, is that, yeah, the pre-drug use for county was higher than pre-drug use for private. So those who went to county tended to have be more hardcore users from the get-go. And so indeed, baseline drug use is related to the type of program you go to, which means it is related to our independent variables. That's one of the two things that has to be true for something to be a confounder. Does baseline drug use, is it related to subsequent drug use? That's the next thing we need to test. So to test if indeed baseline drug use is related to the DV, that is subsequent drug use, we're going to do a bunch of correlations. So we're going to correlate the independent variable, dependent variable, and covariate. We're basically just going to throw everything into correlations at one time. This answers first question, which is what we just answered. Is the covariate related to the IV? So remember, what we just found is that baseline drug use was higher for those who went to the county program than the private program. Here's our correlation matrix. We got to find the correlation between the covariate, that is pre-programmed drug use, and our independent variable. There it is. It's negative 0.588. Significance. There it is. Significance is less than 0.05. That's our test. And indeed, 0.001 is less than 0.05. There is a significant negative correlation between pre-existing or baseline drug use and the group you're in. Well, we know that negative correlations mean that as one variable goes up, the other one tends to go down. So as program goes up, baseline drug use tended to go down. What does going up on program mean? Well, program can only have two levels. You're either a 1 or you're a 2. So to be going up on program means you're 2, which means you're private. So as program goes up, that is private. Baseline drug use tended to go down because it's a negative correlation. That is a complicated way of saying that those who went to private tended to have lower baseline drug use. So yes, or flip of that is higher pre-drug use associated with county program, which is just the opposite of what I said. But it works. So the second part of it, so we knew this anyway, right? We knew that those who went to county tended to have higher drug use, or the flip of that is those who go to private tend to have lower drug use. Second part, though, is that covariate related to the dependent variable. This is the second piece we needed to establish to be able to say, yes, baseline drug use is a potential covariate. We already know that county is higher baseline, but is your prior drug use related to your subsequent drug use? That's where the correlation between the covariate and the dependent variable comes into effect. There it is. It is a strong correlation. Geez. So it's 0.821. Sig is positive. The sig for that is less than 0.05. So indeed, there is a significant positive correlation between pre-drug use, baseline drug use, and post-drug use. Those who, so as one variable goes up, the other one tends to go up, or vice versa. As one goes down, the other tends to go down. They tend to move in the same direction. So this tells us basically that yes, those who have higher pre or baseline drug use tend to have higher subsequent drug use, which is a complicated way of saying our covariate is related to our dependent variable. So this is a potential confounder. So Cookie Monster wants to know why the heck we're doing this. What is all this stuff? Why did we do all this correlation and t-test stuff? Well, we were trying to establish that our covariate, whether it was indeed a potential confounder. We found that the covariate was associated with both the dependent variable, that is the correlation. We found that those who had higher baseline drug use tended to have higher post-treatment drug use. And also that the covariate was related to the independent variable. We did an independent samples t-test and found that those who went to county tended to have higher baseline drug use. So we got ourselves a confounder here. So pre or baseline drug use is indeed a potential confounder of the effects of treatment on post-treatment drug use. That is those, because how you started differed between programs, and it's related to where you end up. It is a potential confounder that makes it a good candidate for a covariate in the study, because your pre-existing differences in drug use could explain differences in the perceived effectiveness of the two types of treatment programs. So now let's go ahead and redo that ANCOVA, but throw in baseline drug use as a covariate and see what sorts of results we have. So we're going to do a true ANCOVA now, where we adjust the means for the covariate and then see if the two treatment programs differ. That is, whether there's a difference on the independent variable after adjusting. So the question here is, does post-treatment drug use of county versus private treatment programs differ after adjusting for pre-existing differences in the drug use of people who go to the two different programs? That is our covariate. Let's first do a test of our homogeneity variances assumption. So there it is. There's Levine's test, pops right out of SPSS. This is one of those ones where you don't want your sig to be greater than 0.05 because you don't want to violate the homogeneity of variances assumption. So is the homogeneity of variances assumption met? We're going to look at the sig for Levine's. Our decision rule: if Levine's sig is greater than 0.05, the homogeneity assumption is met. If Levine's sig is less than 0.05, nope, got a problem. We have not met the assumption of homogeneity of variances. Should that happen, one thing you can do is use a tighter alpha level for your ANCOVA or your independent variable effects. So maybe 0.01 instead of 0.05 to try to account for excuse me, the type 1 error inflation that occurs as a result of violating the assumption. But in this case, our sig for our Levine's is 0.541, which is greater than 0.05, so the homogeneity of variances assumption is met. So that is the spread of scores, spread of post-treatment drug use scores is about the same for those who went to county and those who went to private. So let's get to the interesting parts. That was our Levine's. Now let's answer the question, all right, we adjusted for pre-treatment or baseline drug use. Did that adjustment meaningfully make a change to the dependent variable means? That is, did the covariate meaningfully adjust the dependent variable? If we adjust for baseline, does it meaningfully adjust the means for subsequent usage? Here's our ANCOVA summary table. Note there's a row there for pre-use, program, and error. Crossed out the stuff you don't need to care about. And we want to know if we can reject the null that pre-treatment use does not meaningfully adjust post-treatment drug use. Look at that row for the covariate, in this case pre-use. And the sig is less than 0.05 indeed. So we are going to reject that null. And we are going to say that, yeah, indeed, pre or baseline drug use did meaningfully adjust the post-treatment drug use means, and so it was a meaningful adjustment done by the covariate. All right, here's the big question. So we know that the covariate did meaningfully adjust the means, but does post-treatment drug use differ between persons attending county versus private programs after adjusting for differences in their drug use that existed before treatment? That is, after adjusting for baseline drug use. Is there a difference between those who go to county and those who go to private in their subsequent drug use? Same table as before, we're just going to look at a different row. Can we reject the null that county and private treatment program attendees do not differ in post-treatment drug use? That's our null. There's our row. Sig there for program, our independent variable, is 0.243, our decision rule. If sig's less than 0.05, we're going to reject that null. There's still a difference, because remember in the ANCOVA there was a difference. If sig's less than 0.05 for the independent variable, we're going to reject the null. That is, there's a difference between post-treatment drug use for those who went to county and those who went to private, which is another way of saying there's difference in effectiveness. If sig's greater than 0.05, we're going to retain the null. There's no difference in the post-treatment drug use of those who went to county and those who went to private programs. In this case, our sig is 0.243 is greater than 0.05, so we are going to retain the null. That is, after we adjust for baseline levels of drug use, there's no significant difference between those who go to county and those who go to private and their post-treatment drug use. So remember in the ANCOVA, this was statistically significant. When we adjust in the ANCOVA for their baseline drug use, we find that the two programs are equally as effective or ineffective, however you want to look at it, but there's no difference in the subsequent drug use of those who go to the two different programs. So let's take a look at the unadjusted and adjusted means. I told you that it's typical that you would actually do a figure of these. So the unadjusted means are the crude means without any covariate adjustment. They come from the descriptives table. There they are, the 5.36 and the 3.52. Those are means without any adjustment. Then there's the adjusted means. Those come from the estimated marginal means table. They are the means after you statistically equalize the groups on the covariate. That is, after you adjust for the covariate. there's your estimated marginal means, and you can see they both change. So the county went from 5.36 to 4.64. The private went from 3.52, moved up to 4.24. So these means reflect the impact of the covariates, that is the adjustments of the covariates after you take out any variability in the dependent variable that can be accounted for by the pre-existing drug use levels. What are the means? And it looks like they got closer. So what's our conclusion here? After we adjust for pre-existing differences in drug use, attendees of county and private treatment programs, and note the means there are the adjusted means, 4.64 and 4.24, do not significantly differ on their post-treatment drug use. So the plots, if you're visual like I am, there's typically a plot in ANCOVA APA results sections that show the effect of the adjustment. So you're going to show the unadjusted means and the adjusted means for each level of the independent variable. There it is. So county programs are orange and turquoise is private. And you can see there's a big difference between the bars on the left there, the unadjusted means. The means got closer together on the right, the adjusted means. So after we accounted for their baseline drug use levels, the means got closer together. One went down, the other one up. That's what happened. So the covariate adjusted means are clearly closer together than the unadjusted means. And that's why when we did ANOVA, regular old ANOVA on the means on the left, you get a significant difference. But when you do ANCOVA, that is where you adjust for baseline levels of drug use, you get the means on the right, and you find, nope, those aren't meaningfully different. And that just sort of visually shows you big difference, little difference after adjusting. So what's going on here? Well, the confounding by the fact that the more hardcore users go to county was making the groups look different on their post-treatment drug use, when really it was confounding by the fact that you've got more hardcore users going to county than fancy private programs. And I show you the different sets of means down there at the bottom. County means subsequent drug use means went from 5.36 to 4.64. That went down. Private went from 3.52 up to 4.24. The means ended up getting closer together, and those two subsequent means there, 4.64, 4.24, are the adjusted means that are shown on the right in the graph. 5.36, 3.52 are the unadjusted means that are shown on the left side of this graph. So it's also customary to present the adjusted and unadjusted means in a table along with their standard deviations. This is where we have to convert those standard errors for the adjusted means to standard deviations using your calculator or phone. Here is the table that you want to create. You've got a row for unadjusted means, a row for adjusted means, and columns for each type of program. Where do the unadjusted means come from? Well, they come straight from the descriptives table. That's where you can get the means and standard deviations just as they are, just round them to 2 decimals. Now the adjusted means, that is adjusted for baseline or pre-treatment drug use levels, the means themselves are really easy. If you request the estimated marginal means, pop them right in there. But note the next column over is called STD error, that's standard error. Well, crap, that doesn't help us. We need standard deviations, so what do? Well, we got to convert these standard errors to standard deviations for those adjusted means using the following formula. Standard deviation for a particular level of the independent variable is equal to the standard error times the square root of n, where n is number of participants in that group. Now note there's 25 in each group. So for county, it's 0.214 times the square root of 25, which is 0.214 times 5, or 1.07. Similarly, and that goes there in the table. Similarly, for the private program, it's 0.214 times the square root of 25, because there's 25 who went to private in this study, and you end up with the same standard deviation, 1.07. So not too bad. Measure of effect size, so is eta squared It's sum of squares for the independent variable divided by sum of squares total, where that sum of squares total you have to hand calculate. It's sum of squares for the IV plus the sum of squares for error. Here's our ANCOVA summary table. There's the formula all filled in. We've got our sum of squares for the independent variable is 1.264. Sum of squares total on the bottom there is the sum of squares for the IV again, 1.264, plus sum of squares for error, which is 42.558. So you add those together, you get 43.822. And if you do the 1.264 divided by 43.822, you get about 0.029, which is about 3% of the variability, or 0.03 proportion. That is your eta squared. So finally, your ANCOVA summary table. All you got to do is copy stuff from SPSS, from the output table you get there. This thing we've been looking at, those rows, to an APA format table in your APA write-up. So there's BARF and NESI, and everything just sort of slides in there. Pre-use would go on the prior use table. Program would be program. Just throw them in there. You got to add your eta squared, of course. However, the total row, you just have to sum the things above it. So there is no row that gives you that number that SPSS puts out. So you just sum 49.44, 1.26, and 42.56. Apparently that gets you 93.26. And if you sum those degrees of freedom, 1, 1, and 47, you get 49. So do note you do have to do that by hand. So that is ANCOVA.